Hello, everyone. I'm very pleased to introduce our speaker for today, Dr. Issam El Naqa. He is the founding chair of the Department of Machine Learning at Moffitt Cancer Center and professor of oncological science in Tampa, Florida. He is a certified medical physicist by the American Board of Radiology, and he is a recognized authority in the fields of machine learning, data analytics, and oncology outcomes modeling, and has published extensively in these areas with more than 270 peer-reviewed journal publications, and 5 edited textbooks. He has been a member and fellow of several academic and professional societies, including the AAPM, the IEEE, the IIMBE, and the BIR. His research has been funded by several federal and private grants in Canada and the US, and served on national and international study sections. He acts as a peer reviewer and editorial board member for several leading international journals in his areas of expertise, and he's also the reason why I got involved in the radiation biology, and he was my boss for several years from 2011 to 2015, and I'm very grateful for his input in my life, let's say. Welcome today, Dr. Issam, and it's an honor to have you here.
Thank you very much, Norma. I'm very grateful for the opportunity and for the kind introduction you gave me. And if I was doing a favor to radiation biologists, was hiring you personally. It's truly an honor to be talking to you, and thank you, Pete, for organizing this and everyone who took the time to join in. Let me just get talking about AI, and you've probably seen how AI evolved over the time. I mean, we started to talk about these things when I was at McGill many years ago, and since then, a lot of things had changed. Actually, AI became a focal point for many applications. So these are my disclosures, and talk about our learning objectives for today. Talk about outcome modeling in general in radiotherapy applications, how AI can play an important role in this area, some of the challenges associated with it, and some vision towards the futures that probably cherish your input on. The picture you see in the background is actually, I gave this information to ChatGPT and asked it to generate a picture, and this is what it came up with. Now, let me start by a quick disclaimer. This is a statement actually by Fermi, and he's talking about his friend Van Neumann, the person who came up with the microprocessor type systems. And I mean, they're talking about data regression and data fitting, and pretty much saying with 4 parameters, you can fit an element, and with 5, you can make it wiggle. And practically, this is what we do in AI. I mean, 5 parameters would be a joke when you're talking about AI. We're talking about hundreds, thousands, millions, billions. And I mean, if you follow some of the latest news, people are talking about trillion type of parameters. This is a textbook. Actually started working on it when I was at McGill. It's Guide to Outcome Modeling that has many aspects related to what I'm going to discuss today, although the architecture of AI has evolved significantly, but the fundamentals remain quite similar. So outcome modeling is the use of math, physics, and biology and computer science to study behavior of tissue response. In this case, we're talking about radiotherapies. And the idea is to make predictions about the patient outcomes, whether it's the tumor response, normal tissue side effects. Sometimes we use PROs and quality of life in response to the varying treatment conditions. So the idea of outcome modeling is to take the real world where you are making clinical observations and map it to a mathematical world using techniques based on AI or other type approaches, making predictions based on these techniques, then going through a feedback loop in order to improve the performance of that mathematical models so that it can match as much as possible what we are observing in the clinical practice. Having that wealth of information, then you can start personalizing treatments and making decisions accordingly. Now, just a quick background. It might be all familiar material for many of you. But just to level the stage, outcome models in radiotherapy actually date back to the 1930s. There are 2 main clinical endpoints. One is tumor control probability, TCP, and the other one is normal tissue complication probability. This diagram you see here, that these are typically represented by these sigmoid shape when you are looking at a single variable like those here. Like this is the TCP and NTCP. And ideally, you would like to have these two as much separated from each other. When you are doing optimization, you create a function like, look, that this one, which is, for example, I want to maximize the TCP multiplied by 1 minus NTCP, which is called the P plus type function. And the idea, I would like to have a plan, a treatment plan that I can develop that has 50% tumor control or more, and toxicity does not exceed 5%. Now, the question is, why do I need AI for modeling response? This is an example here. This is for Quantic that there's probably now, 14 years ago. And what you are seeing is trying to predict inflammation of the lung, radiation pneumonitis, an area that worked extensively with Norma when I was at McGill. And we see these are the predictions from multiple institutions. And you can see most of them having similar type of predictions. And if you are looking at the changes in dose that leads to toxicity, what you are observing here is the change itself is less than 2% per gray. Which tells you that these models are actually more general of the population rather than the individuals, because we know a change in 1 gray is a significant impact versus what we are observing with these population type models. So if I take a population type a model like this, which is what a classical radiobiology would allow me to do, and I would like to decipher the individual responses out of these populations, I need more information in addition to the dose that are reflection of the individuals that are being treated. And this is a graphics from Dr. Tenhaken actually demonstrating these type of effects. So population-based model tends to be shallow. What we aim to achieve with AI and other techniques is making them steeper so we can apply them to the individuals or to try to recover the individual from the collective collection of the population itself. How we do that? Using data, and we need more information that are patient specific, which is currently actually becoming abundant, and that's a main driver for AI. The sources of information are shown here: tissue, blood, saliva. The pieces of information could be, the genetics information, the protein profiles, in addition to the dosimetry, which talked about earlier, or the imaging information. You would like to have labels, although with more advances of AI, probably these are also can be resolved from the data currently. Now, we cannot have enough information to do modeling or outcome modeling. This is an example, actually, work started at McGill with Susanna Heckling, looking at radiation acoustics and whether we can visualize the dose in real time. And moving forward in that type of work at Michigan, we have developed these custom-made ultrasound transducers that allow us to visualize radiation in real time in vivo type models. This is a rabbit models, and we are currently having a clinical study actually that is still ongoing. They are doing it in liver cancers. These are the planned dose here, and you can see what you are able to visualize of the radiation while it is being delivered in real time, which is quite intriguing and would have a tremendous impact on the way we can do adaptive therapy, all feed better outcome models about the actual delivered dose versus the planned dose itself. Now, radiological outcome modeling can go in two directions. It can go a bottom up, which is going from basic principles, and this can be done with Monte Carlo type techniques or DNA track structure type approaches, which actually did some of that work at McGill too, or going more integrative approach top down, where you are looking at the clinical endpoint and trying to develop an integration model from all available information for that. Now, in terms of the basic principle of radiobiology, I mean, these are valuable pieces of information remain. So this is looking at in vitro cell culture assays, and some of the initial models, as you can see, these are multi-target single hit or the linear quadratic model, which we still use with its these parameters, alpha, beta, parameters. Then there are these different principles of radiation biology that are important, that whatever we are developing from AI still conform within that framework. Now, for normal tissue complications, organizational structure still plays a role. Now, there are these concepts of functional subunit, whether they are parallel or serials, affecting which part of the DVH is actually responsible for the response itself. The reason I'm bringing this information, because whatever we are doing with AI or more advanced data-driven need also to match what we can do with classical radiobiology and use that pieces of information as prior knowledge that can allow us to constrain these type of techniques. And I'll come back to this later when discussing more deeply into AI technologies. So in the integrative radiotherapy response modeling, I mentioned we'll talk about TCP and NTCP. And the consensus currently that these are multifactorials, meaning I'm going to combine the dose, which is the physical piece of information, with the genetics, and that's the field of radiogenomics, or combine it with the imaging information, and that's the field of radiomics, all together. Going back to the example I was showing about the original TCP and NTCP models, are creating this P plus function. Currently, instead of just using those, we are creating pieces of information that is dependent on all the different variables that we can have access to, whether that is the imaging, genetics, clinical, and so on. Now, traditionally, there are analytical models that uses these pieces of information. So looking at the radiation pneumonitis, again, you can do maximum likelihood estimates. And this is the shape of the curve that comes up from this kind of design. It's kind of a fixed shape that you are assuming it takes this format. Now, in AI, you can think I have hundreds or thousands of these that I'm combining together to create a nonlinear behavior, depending on the interactions of the different variables. This is a software tool that we developed many years ago that can allow us to do these traditional techniques on some of the basic data-driven type techniques. And this is one example predicting NTCP models for radiation pneumonitis. Now, this is work done with undergraduate at McGill, who actually completed his Ph.D. later on at Oxford. And what he did working with Norma and the team at the time is combining those with genetic information for predicting rectal bleeding and prostate cancer. So if you take this example, this is a pure dissymmetric model, the blue line. You add a genetic number to it, the copy number variation. You can see, for example, for the 50 gray, if the original prediction was around 0.05, when you add the dissymmetric variables, it increased actually significantly. It's practically double for this population. This is when you do it using data-driven type techniques. They're looking at the data fit, but it is similar in terms of what you are trying to achieve, which is personalizing the decision, the prediction to the individual rather than the population by including their different genetic factors in this case. Now, you can use the AI, this is the principal component analysis, to look at the relationship between the dissymmetry and the genetic. And you see there are orthogonal relationship indicating that these variables are actually complementary to each other. So this is one use of AI early on. Now, it's not only looking at dose and genetic, you can look at the protein profile, combine it with imaging type information. This is looking at prediction of liver function by combining DC MRI with the protein profile with the dosimetry. So if you look at the dose only, this is the functional model you see here, and you look at changes in cytokines, in this case happen to be TGF-beta, you can see there's a significant difference what's happening looking at a physical variable only or combining that physical variable with the protein profile, indicating how important that this patient-specific information, whether it is the genetics or the protein, in improving prediction performance. Now, another quote, this is with Ron Fisher, is considered a godfather for statistical modeling. And this is what he's saying, and direct quote, "No one who is familiar with mathematical advances in other fields or a range of special biological condition will be considered summing all this information in a single mathematical formula, however complex. And he's absolutely right. I mean, if you are using direct mathematical type models, you will not be able to capture all these pieces of information. And that's we use AI, which is like an ensemble or all these different models and can allow nonlinear complex interactions to be captured. Now, there are many different flavors of machine learning techniques. There are the generative models, which are coming fast and furious nowadays back again. Some of the basics ones are the Bayesian networks or the Markov type models. More commonly used are the discriminant type models like the neural networks, the CNN, random forest support vector machine. The difference between them, this is a black box input-output mapping, and this is more transparent or semi-transparent relationship. And we are currently moving actually in this direction with many of the newer AI development. Just a quick reminder about deep versus conventional machine learning. In conventional machine learning, you do feature extraction, feature selection, then you relate that to input-output. In deep learning, you combine all these steps into one. A very common example is this convolutional neural network looking at PET-CT analysis. You see a tumor here. These convolutional layers, again, it's doing the same function. It's just combined in a single framework, doing feature extraction. The pooling layer are doing feature selections. Then you can pick up your favorite classifier, which happen to be a multi-layer perceptron, to doing the classification between being malignant or being benign. Now, there are many architectures of neural networks. This is some examples of these, the MLP1, which is the basic architecture, original one, the CNN, which is more applications in imaging typically, recurrent neural networks, which look at times and sequences, which is the precursor also for using transformer. This gentleman here is Hopfield, and he won a Nobel Prize in chemistry recently. Autoencoder, is a way to data representation or data compressions, and GANs is one of those breakthrough in AI where you are pitting two neural networks against each other to generate synthetic data. Now, most of the talk currently about generative AI and foundation models. The idea of foundation models is models of models. Practically, you take all the pieces of information you have, you train it, you generate this big model, then you adapt it for the different tasks instead of defining a model for each individual task. You can project that idea on medical applications, and you generate these foundation models that could be applied for healthcare, biomedicine. This could be patient-facing or physician-facing in order to make individualized decisions, whether on prediction of a response or any kind of application in the healthcare industry. Now, these are based on the designs of the transformer, which is becoming the working horse for many of the Gen AI applications, where you are looking at encoder-decoder type design or encoder-only, decoder-only designs. And with these type of designs, the main difference between this one and the generic recurrent neural networks is recognizing that positioning can be actually made explicitly rather than implicitly, and also adding the attention. And actually, that was the title of that transformer paper, that by creating attention mechanism, which is more mimicking of human behavior, you would be able to achieve better performance. That's drove the whole industry of large language models for natural language processing type applications. Probably many of you familiar with the transformers, the different GPTs, including GPT-3 or GPT-4, the chat GPT, and Google's been talking for a while about this type of trillion type parameters. So it's becoming a game, how many different parameters. And if you remember what Fermi said about 4, I mean, look at this one, it's 1.6 trillion type parameters. Now, whether we need all these parameters for application in medicine, that's an open question on its own. Now, some of the applications that are of interest is extracting pieces of imaging information. We can do it with the feature base, which is the shallow learning approach, or you can do it with the featureless base, which is more of a deep learning type approach. This is a radiomics model developed by Lise Wei in Michigan, looking at prediction of survival post SBRT treatment in lung cancer. And you can see what's happening in this model. It's actually not only using the deep learning, but also using classical radiomics features and clinical features. Because with limited data sets, you need more pieces of information. So that's why these intermediate steps. What's interesting about the model, these kind of images that you see here about where the model actually highlighted areas of interest. These are using GradCam, just as looking at activations of the different layers. It highlighted areas in the liver that seemingly were drivers. Initially, we couldn't recognize these, but when presented to our radiologists, they seemed to present areas of cirrhosis. That was the main driver for prediction in that population. Now, staying with the liver application, but in this case, looking at liver functions, this is data from a clinical protocol where MRI images, like the example I showed previously, were collected pre-treatment and around the decision point after 3 fractions during the course of treatments. However, this is very expensive and also added more effort on the treating team as well as the patient itself. And using some of the technologies of AI, the question was, can we, using this pre-treatment information, we know what the baseline MRI image was, and we know what the dosimetry that's going to be delivered. Can we predict what this image look like and look else as well as the function of the liver, the NTCP type model? And the answer to that question was actually maybe. As good as these GANs are, they are also require sufficient sample size. For if the liver function was good, the model actually performed very well and the predictions were relatively accurate. But if the liver function was bad, they called child-Pugh C in this case, the performance of the image prediction was actually very bad, but the relative prediction of the NTCP itself remained relatively decent. So despite having a bad image prediction, the model was still able to do good prediction. It's looking at the difference between baseline and what's happening in mid-treatment. Now, in the era of multi-omics, you are combining multiple pieces of information. So you have the imaging information, you can use CNN, you have the vector information like genetics or a protein profile. You are using these autoencoder type designs that you can use it for predicting radiation pneumonitis or using the imaging information with the dosimetry to predict local control. And this is something these models can do that with the classical mathematical techniques or statistics you can do. You can predict multiple endpoint at the same time, as well as predict time to event using nonlinearity and variable interaction as part of the model designs. This is looking at the C index, which is, you can think about it as an average you see in terms of performance using these neural networks applications. You can see it's actually above 0.7. This is an independent validation, with still relatively decent for radiation pneumonitis and local control, but it's definitely much better than the baseline of radiobiological model looking at the symmetry only. Nevertheless, there are still much room for improvements, and probably adding more information to these network architecture would be helpful in improving these predictions. Now, once you have these predictive models, the other question is what we can do with them. This is an example looking at predicting local control or local failure in patients who received SBRT in lung cancer. As you know, the success rate in this case is about 90%, but there is this 10% of patients that seems to fail consistently. And the question, what can we do about that? Now, if you can identify this cohort of patients, you can use explainable AI techniques like the GradCam mentioned earlier to look at what are the issues with these patients. And you can see, looking at the dosimetry information here, it's about the margins and the overlap with surrounding organs at risk that is probably need to be boosted for this population. So instead of using 100 BD, you probably need to go 104 for this specific subpopulation in order to overcome the risk of failure. So this is an actionable type approach, not only doing the prediction, but also acting on it for treatment planning practices. That's kind of a step forward of what we can do with these techniques. Some of the other work that we can do with this is actually changing schedule, the fractionations themselves. This is a clinical study where they were collecting pre-treatment PET and mid-treatment PET in addition to other genetics and molecular variables. And the question is, if I compare dose escalation based on this kind of model to baseline, like 2 gray per fraction, can I improve outcomes? And the answer could be yes, but depending on the conditions of the patients. As you can see with the dotted line representing the clinical practice, in some cases, yes, which is the green dot, and some cases, no, which are the red dot, and the orange represents similar to clinical practice. The other question is, can I use machine learning techniques to mimic what a physician is actually doing in terms of these type of predictions? And the answer is yes, but we need more data. And that's where you get the GAN algorithm in order to generate more synthetic data to enrich the environment. You need also to identify the environment when applying the methods. And you need an algorithm that is able to search all the possible options and give you a prediction what would be optimal under which type of condition. And these are called deep Q learning neural network. This is a form of reinforcement learning type techniques. Now, in terms of the performance itself, you can see there is quite a good follow. These are ordered chronologically between the prediction of the algorithm and the clinical practice. But if you fast forward here, you can see the clinicians actually making better decisions than the more conservative algorithms, but sometimes it's actually making failed decisions. And that's tell you that the algorithm was able to learn the protocol, but humans actually do not necessarily follow the protocol. They use their creativity, and they can perform better than a pre-trained algorithm, like in this case. Now, clinical translation is a key aspect of what we do in our practice, and that's why we develop these software tools that would allow us to share this information with the community. So this software tool allow you to do these model predictions and optimization of decision making that I mentioned earlier. But another important aspect is looking at the human factor associated with AI implementation. So if I give a software AI tools, how expert physicians are going to use it. So we conducted an interaction, a human AI interaction study looking at this specific case when an AI is making decision about the schedule, how the physician is going to take it or not, and what they are going to do with this piece of information, and what are the factors that can drive such decision. And the answer to this question turns out not to be easy. It actually varies by evaluators, their backgrounds, their preconceived ideas about AI. It varies by the different sites. So these are results from the lung, and these are results from the liver. They actually different cases have different responses. And it also varies from case to case. It's very hard to get quite a consistent performance. But generally speaking, a good number of the physicians, in this case were about 15, actually cherished the AI decision. Some of them act on it. Some of them is actually totally rejected, although the majority felt there is a value in its utilization. Now, there are many challenges associated with these multi-omics type modeling aspects, and these are some of the papers that summarizes some of these issues. I want to highlight a couple of papers here. One looking at the ethics associated with that, because it's actually a major factor in some of these decisions and how we have a successful and safe AI in the future. It's written actually by a bioethics lawyer, the first author, and the other one about the aspects related to clinical translation of AI into clinical practice and how we can facilitate that process so we can reap the benefits of these AI techniques. Now, there are checklists all across the board when you go to AI applications for outcome modeling. This is actually by Suna Choi in collaboration with different colleagues, looking at the different aspects that can result in a good AI models, particularly looking at issues related to systematic bias and adding safeguards to improve performance when you are making decisions on something that you don't see. This is outcome modeling. There is no grounded truth when you are making that decision. Now, some of the ethical challenges you've probably seen these examples, and there are many other examples related to data access, which remain always an issue, and it's becoming more and more complicated by new regulations. Now, there is one way of doing AI, which is bringing all the data together in one place, or using a federated type approach where the data stays in the institutions and the algorithm visit these different data elements, which is the federated learning approach, which is gaining more steam nowadays as a way to overcome many of the issues associated with data sharing and building centralized data type applications. Now, another issue associated with AI application is data imbalance. And when you are looking, for example, a prediction of local control in lung SBRT, it's, as I said, success rate is 90%, which is fantastic for the patient, not good for the modeling, but the failure rate is 10%. So there is no shortage of these different augmentation techniques. And this is an example where Dennis Dudas has actually looked at these different approaches for synthetic data generation. And this stable diffusion denoising technique seems to provide the best performance. And this technology is actually widely used for synthetic image generation currently in the field. There is a conundrum between accuracy and interpretability. Deep learning techniques tends to be quite accurate, but less interpretable than, for example, decision trees or Bayesian network I mentioned earlier. But now there is more convergence of developing techniques and technologies that actually bring the two together to this ideal approach quadrants that we hope to achieve one day. There are many visualization techniques for looking at explainable AI. Which work very well when you are dealing with images, but when you start combining images with other pieces of information, say genetics, protein profile, it becomes a little bit more complicated because it's not a visual exercise anymore, but it requires prior knowledge or functional analysis to prove that this is actually working. Again, we are developing a software tool. This is in collaboration with NIH and other university to facilitate the availability of these tools for clinical or research use. This is focusing on two aspects I mentioned earlier: explainable AI using visualization tool like shown here, as well as federated learning in a multi-institutional type setting. Now, there are also ideas and actually stems from some of the medical association rejecting idea that autonomous AI can actually have its own world to define decisions, that having a human in the loop would be an important aspect. So the area is called IA instead of AI, so intelligence augmentation or a human in the loop. And the premise here, if you add a computer to a brain, it's going to be better than a brain only, assuming the prediction of the computer are actually positive. So take an example of Bayesian network analysis as shown here, predicting again local control and radiation pneumonitis. When you show this one to our physician, they were not happy because some of their favorite variables were not part of the design itself. And some of these variables are variables you might relate and should be there. We'll talk about lung cancer. Probably we should add smoking, we should add chemo, the size of the tumor, among many other variables. So when we did the analysis on this data, what we noticed, the prediction did not change, but the confidence intervals actually were tighter. This is an indication that the generalizability or its performance on out-of-sample data can potentially be better. So the human in the loop actually will have a significant application in areas, like I mentioned earlier, looking at SBRT, for example, for local control. This is in liver in this case, where the prediction, again, the event rate is only 10% failure. And here asking the human to predict what will happen in this case. And you can see they go above random, which is about 60%. Do data analysis only, it's about 80%. But if you combine both together, you are not only improving generalizability, but also you are improving the prediction performance, about 86%, which is quite significant. That's an indicator that there is more information that the expert have that does not were not properly represented in the data or were insufficient in the data, particularly when you have a skewed event rate, like in this case of local control in liver cancer. I want to switch gear talking about the futures, at least the futures I perceive it in the machine learning world. I think the combination of physics and machine learning would be the main driver for years to come. And just to quote Richard Feynman talking about plenty of room on the bottom. And the idea, what is the motivation for physics-assisted machine learning, or what's called QML? Well, current classical computing is actually reaching a bottleneck. And even if you, not only on the computing side, but also on the algorithmic side, there are arguments that deep learning is reaching a plateau. But there are other aspects that these type of techniques, these are stochastic technique in nature, that they can deal with uncertainty in the data. And we talked about the bias aspects, uncertainty in the model and model selection architectures, and probably how we can balance bias and variance. Bias ability to fit the model, variance how it performs on out of sample data. And there is this theoretical framework that you can an optimal model is actually that balances bias and variance. This is actually can be derived for simple architecture, like an SVM with a linear kernel, you can derive this, or with a polynomial even. But if you start talking about neural networks that exceed 2 layers, what that deep learning is, then that kind of theoretical framework does not actually work very well. So how this combination of quantum and machine learning can work? Well, let's look at the options that are available. The data is classical. This is what we currently encounter with medical applications, but the algorithm could be quantum. Or the data could be made quantum, and the application, the algorithm could be classical or any or a quantum. What we care about for medical applications, the data is always classical, and we can use a combination of classical and quantum, and I will show examples of that. Now, there are three specific aspects that I think would be very valuable to use that quantum can lend itself for improved machine learning application. Data representation. The first one, in typical data representation, you are dealing with zeros and ones. With quantum, you are dealing with the qubit, which allow you to actually combine informations between 0 and 1. This is the block circuit, which is, as you can see, this is the data representation that include an amplitude as well as phase information too. Now, there are also other properties that are beneficial for data representation. The superposition principles that actually probably shown here, and the parallelism, but also dealing with highly correlated data. This is one of the major pitfalls when you are dealing with regression models and taking to machine learning models. When the data, there's high linear collinearities in the data or nonlinear collinearities, it's created problems for these models to decipher what is actually real signal and what is actually noise, and probably principles like correlative entanglements can help with that. When dealing with dynamic system, trying to predict things over time, the Schrödinger equation is a great objective function to use. I'll show example of that, as well as the search algorithm. I talk about looking at optimal prescription or optimal schedules. One of alternative for the Q-learning is the Groover search algorithm, which relies on amplitude amplifications. And there are other aspects related to phase estimation that could potentially improve performance. These are some examples that demonstrate the value of quantum machine learning versus using machine learning. One is the speed up, which is the classical example for treatment planning applications. And in this case, you can get a similar performance to what you get in a classicals, but in a shorter type of time. Another example, when you're looking at longitudinal data, is using these MR images, try to predict disability. In this case, happen to be multiple sclerosis on these patients using MRI, where you can get a good performance. In my opinion, the best applications and when you are dealing with adaptive, whether it's geometric adaptive type applications, which are looking at changes during cone beam CT and try to predict the setups. And in this case, it's actually using the Schrodinger equation and try to predict what would be the Hamiltonian in this case and try to get a performance. However, the main areas that I think QML can outperform classical machine learning techniques would happen in making decision under conditions of uncertainty. Just to dig deeper into this application, I mentioned earlier the reinforcement learning application with DPQN, and here we're creating a quantum deep reinforcement learning algorithms. Now, in order to train algorithms on a limited data set, these basic principles I mentioned earlier about predicting TCP and NTCP using these sigmoidal type functions actually becomes handy when the data is limited. It's create constraints on the shape of the reward function that is actually part of the design of the reinforcement learning algorithms, as well as what's the expectation associated with that environment. Now, mind you, the environment itself is an underrepresentation of the actual patient environment. So it's actually a noisy type environment, and somebody's going to make a decision based on that. So application, for example, of the Groover algorithm, which is the search quantum learning algorithm, what will happen if you apply it directly under a noisy conditions, trying to do this amplification, searching for a number like this. This is what happens when you're dealing with noise, when you try to add more realistic noise to the data. And imagine trying to project that on a decision making by a human where all these variables are actually noisy. Becomes a big problematic problem. And that's why we actually changed the design of the original Groover algorithm, changed this NOT gate into more parametric versions of it in order to enable dealing with noise, which is, I think, that was the major contribution of that paper. And of course, they did the simulations and testing it on one of the open source quantum computer, which is the IBM Q1, as shown here. Now, in terms of the results, looking at the data set we have, when you are looking at root mean square error, I mean the regression type aspects of the algorithms, you don't see any difference between all the three of them, whether you are using simulated, you are using the real quantum, or even using the classical machine learning algorithms. In the validation, there's some, a little advantage for using the quantum on real computers. But when you ask the question differently and asking about decision making itself, when did the computer make a good decision or a bad decision? Well, can I escalate the dose or cannot I escalate the dose? If the patient, for example, has an improved local control, or the tumor is responding, you don't want to escalate the dose. I mean, that's kind of natural human behavior. But when the tumor is not responding, you would like to escalate the dose. Then you look at the toxicity. Am I increasing toxicity or reducing toxicity? Now, the yellow here is showing the clinical decisions. Remember, they don't have access to the future. They don't know what will happen eventually to the patient. Neither do the algorithms. But they are learning from the data what could the future be. And you can see all of these algorithms actually outperform the human because they can learn more from the data. But in terms of the decision making, in terms of best performance, you can see actually the quantum start performing better than the classicals because of the uncertainty associated with the data itself, though the difference is still a work in progress. But you can see there is a potential for these ideas to develop better machine learning algorithm that can operate under conditions of uncertainty. Now, as medical physicists, you know more about quality assurance than I do, but also machine learning and AI would require also their own quality assurance, whether we're talking about AI, ML, or quantum computing techniques. This is an article that discusses some of the basic principles that you're all familiar with, whether acceptance testing, commissioning, quality assurance, and developing a program, and trying to project that into the world of AI and AI application. This is still a work in progress. There is still no consensus about how these could be done. There are different checklists and there are different perspectives, but there is more work to be done in that area. So I want to take the next few minutes just to summarize what I was talking about and take questions from you. I think there is a tremendous use for multi-omics data or multimodal data integrating the physics, biology, or the imaging information if you would like to develop better predictive model of tumor control or toxicity. Machine learning algorithms come with different flavors. They vary in their accuracy and interpretability. And the choice of these algorithms is still practically an art more than a science, but it depends on the experience of the person who are using it. There are barriers for applications for personalized medicine, including outcome modeling, but these barriers can be solved. Some of them, the data access using federated learning, interpretation using visualization, at least with the GradCam, behavioral science, including a human as part of the design. And I think there is a lot of opportunity also by combining physics principles, including quantum computing. Collaborations between the different stakeholders are actually essential for the future of this in order to have safer, more beneficial AI application, whether we're talking general biomedicine or radiation oncology type applications. These are some references, some sources for additional software tools. And I end up by this quote by Richard Feynman's, and I'm happy to entertain any questions you may have. Thank you.
Thank you very much, Dr. Issam, for this very interesting talk with a lot of data and a lot of projects. I think you have been very, very productive in the last few years. So we are ready to take some questions. We will start here with the audience at the Glen side. Do you have any?
I see Eve. Eve have a question already.
Yes. Yes.
Hi, Sam. How are you?
How are you, my friend?
Thanks for joining us.
Absolutely pleasure.
So any questions from the audience here? If not, yes.
You come up with some.
Okay. So,
I see Sharon too, of course.
yes.
Hello.
How
Hi,
are you?
hi, Issam. How are you?
Good to see you.
Great talk. I really enjoyed it.
Thank you.
So I have a question about having this type of multimodal data and so data fusion in general. You try to develop some multimodal training for outcome prediction, and it's not always adding another modality that increases the model's performance.
That's correct.
Yeah.
Okay. Okay. The answer is yes and no, right? If you have a good reason to think that adding another pieces of information will improve performance, then we should do that. The other question, how you are going to add it so it is actually add to the power of the model rather than becomes more detrimental. As you know, if you increase the number of variables that you have, you will have a fitting problem here.
Mm-hmm.
And if you reduce the information to a level that you lost that availability of that additional pieces of information, you will have a problem. So we have to balance the two with each other. What are the pieces of information that I need to add and how I'm going to add it? Now we can go technical about, early phase, mid phase, and late phase data fusion and data integration. And this is, I think, an area where you will have expert on this, what this data actually means, expert in the area, in addition to the machine learning person would be very valuable to come up with a recipe for that. There is no single recipe. And you can see what we did with the application, one of the papers is we use this transform type approach, which is people, currently it's worth go with the word embedding because of the LLM stuff. But when you are doing this embedding business, um, you have to be careful that you did introduce the information to the degree that it got diluted. That's kind of where the balance need to happen. And in order to get that balance, there need to be domain knowledge when applying these different.
Exactly. I couldn't agree more with you. But the problem we've had, and I don't want to take time, if students have questions, we should take them first. But for genomic data, for example, where we have tens and thousands of features and very few patients, so the data itself is scarce, but the number of features is enormous. So it's very difficult to get hold of a domain expert, and people at least at McGill, genome people at the Genome Center, they are very busy. So yeah, I totally agree with you, but sometimes to get a domain expert is not that easy.
No, I understand, but it's very important to get buy-in from them, and they would be helpful. We have a bunch of projects. I didn't list them because it's a little bit off topic, that we are working with medical oncologists, immunologists, and people like that. And they ask us to analyze their data and build these models. I'll be very clear from the beginning, Ryan. If you don't collaborate with us, explain how we should use the data. I mean, we are going to repeat what you got from your bioinformatics person. We can do, I mean, we might be able to do a little bit better, but not much if you don't help us build architectures that are informed by your knowledge. And actually, they participate. I mean, and actually, they give us way more advice than makes things actually way much better than if we've done it on our own. And that has been our experience with many of the applications that are relatively new to us, including pathology applications. I mean, pathology images, we've been working with them. They tend to have an amazing amount of information that, I mean, it's very hard to grasp and very large pieces of information. And without having a pathologist involved telling you what they are looking at, it's a super hard training problem. Unless you have infinite number of samples, then you don't worry about that. Until that time, we need expert to help us. And a couple of examples I showed, even simple examples like trying to predict local control in lung or liver. When you ask the expert and tell them what variables I should be using or what variables the model is missing, it's actually add value to the prediction power, which actually I didn't think this was going to happen. I thought we have it all, let them say whatever they want, but actually it was wrong.
No, I agree with you. So for pathology, fortunately, we will start have once per month meeting with the pathology department to present. And
Yeah,
then we
that
have
should be
that
very
with
valuable. Yeah.
head and neck group we have here also once per month. Clinicians and the students, they list their questions and they ask clinicians. So I agree with you that without a multidisciplinary approach, it's difficult to just.
Yeah, it's difficult and maybe waste of time. That's the risk you are taking. You're spending a lot of time and you're not getting the right information to use.
Yeah, I think now we just need this genomic people to be on board as well.
Yeah, that's what it is. Yeah, that's.
Yeah.
I mean, yeah, I actually know some of that people in McGill. They're outstanding. I mean, they're world-class people there.
They are, but I think nowadays everybody wants to collaborate with them, so the time is scarce. That's the problem,
Well,
I think.
sometimes it's hard to deal with celebrity.
Yes.
Issam would know.
Students who have, I know that many of you are doing this type of study, so you can ask your question. Issam is very nice, obviously, as you can see.
Of course,
Juan has a question. Please go ahead.
Hello there. Thank you so much. I was commuting, so I'm outside right now, but it was an amazing talk. I think I've been a good guy, so I might give myself for Christmas the book of outcome prediction. Early in the talk, you mentioned about how you looked at PCA. I mean, you used PCA analysis to look at different modalities and see like they could complement each other. And I'm wondering, because when you want to do a multimodal analysis, I think it's a good idea to do this before even trying to fuse them in a complex manner. So I wanted to ask your opinion on if PCA could be used and how could it be used if it's different modalities, for example, clinical variables and image. Would it be a good idea to, for example, put them into latent space and then apply PCA there?
Well, that's a great question, Juan. Just to be clear, we use PCA for data visualization, right? It's just looking at the relationship. Now, a lot of people, the original application of PCA is data reduction and dimensionality reduction. In the deep learning world, this is kind of becoming not a favorable type approach because you are losing information. But depending, again, it depends on the application and the situation you are dealing with to decide what kind of reduction you need. PCA is a linear reduction. I mean, there are kernel PCAs, there are ICA, there are a lot of ways to do dimensionality reduction. And it depends on the context you are applying your model to, as well as how much samples you have and how you balance the risk losing information in terms of content versus being able to hit the model with the samples you have. So it's kind of becomes a design question that you need to balance the requirement of that you have in order to address it.
I see. Yeah, this makes sense. I will definitely pay attention to that. I also wanted to ask you about Bayesian networks, because I've been thinking that it is possible that, for example, if there's some visual information in a whole slide image, knowing a condition beforehand about the patient, like lifestyle or so on, this condition could be useful. This information could be useful for a better prediction. And so that's why I'm talking about Bayesian networks. So, for example, knowing a priori distribution of information before making a prediction on another modality. I wanted to ask you if this has been an area of research that has been investigated.
In the past, yes. In recent years, I think most people are focusing on graphical neural networks as an alternative for Bayesian networks. But many of the same concepts can be applied still because the graphical neural network will give you the nonlinear behavior, which is the original Bayesian network does not really have. But you are going to lose some of the interpretability of the graphics when you start combining a node. Adding prior knowledge is actually essential part of the Bayesian. It's the conditional probability multiplied by the prior. But I think a hybrid between the two would be useful. And again, it depends on the type of problem you are trying to solve. But there are also Bayesian neural network addition to the graphical neural network type approaches that exist primarily to deal with noise and noise estimation. Uncertainty.
That's interesting. Yeah, I'll definitely look into that. Thank you so much.
Absolutely.
Eugene, you can go ahead.
Thanks, Pete. Hi, Issam. Good to see you.
Hi, great to see you too.
Thanks for the amazing presentation. I have a question regarding the evaluation metrics for your human in the loop slide. Like you first showed the AUC curve for the human prediction, and then the second AUC curve for the machine only prediction, and then them combined. I was just wondering what kind of how did you evaluate what kind of questions did you pose to the humans, and how did you eventually quote unquote combine them and then evaluated them again?
No, that's a great question, Eugene. What we did, this is actually an area that if you want to look it up, it's called credible AI. So, which is practically what happens is that you take pieces of information, you build a predictive model, you show it to your experts. They tell you are missing variable X and Y, we would like to include it. So when you are doing the feature selection or the model building process, you give higher weights. There are different ways of doing it, but you reinforce the variables that they want within the model building process itself. And you can see details about how it was done in these couple of papers. And once you do that, the rest of the evaluation is pure statistical, right? I'm looking at cross-validation predictions, or I'm looking at testing predictions. In these cases where cross-validation within the data set itself.
So for the human only model, so for if I remember correctly, you were predicting failure for that case. Did you ask them? Is it a binary or is it time to event prediction, or did you ask them
binary? These
binary?
were binary.
And you asked them in for example in the two-year frame, do you predict this patient would fail or not? Okay, Thank you for that. And also, in terms of integrating, for example, dosimetry information to your models that has other modalities, what type of dosimetry information would you think would be useful for this integration? Would it be sort of the 3D dose map, or what other dosimetry metrics that?
Well, that's a great question. It depends on the sample size you have. If you have a limited sample size, we go with the DVHs, which is one of the papers. If we have a larger sample size, I mean, that other paper had like 100 something. In the other paper, we had 500 cases. So we use the 3D dose distribution. The 3D dose distribution is much richer information because, I mean, any DVH reduction loses the spatial information, which is very valuable, particularly when you are looking at not only local control, which can be done just within the tumor itself, but you're looking at the relationship between the different organs when you are trying to make decisions. And that's what you would like to have the whole spatial relationship also being represented. Now, not all tumors are spherical. So also that's another piece of information will be lost. I mean, we'll talk about hotspot and other things. There's a lot of information when you are doing with DVH analysis, you lose. But if the sample size is limited, like your colleague was asking, should I do data reduction? Well, the DVH is a normal data reduction because physicians use it anyway.
Right. Okay. Thank you very much. Thanks again for a great talk.
Absolutely. Pleasure.
All right. So we're at the 1 hour, unless there's any other really hot questions. With that, thank you so much, Issam. This was a very nice talk. Nice to see you again.
Likewise. Thank you very much, Pete, Norma, and everyone. I'd like to seeing everyone. And yeah, it's totally a pleasure. And I look forward to seeing you in person next time. Take care.
Yes,
Have a
that would
great
be
day.
great.
Take
Thank you
care.
so
Bye.
much.
Absolutely.